Hello everyone. Today we will talk about the life of a cell. In this detailed scheme here, we differentiate two main situations in a cell's life. The green period that belongs to what it's called interphase, and the yellow one here that represents the division of a cell into daughter cells. Interphase is the phase that takes place between two cell divisions. During this time, the cell functions as it is meant to do. If we were to represent a cell during interphase, it will look like this. Well, it will be much complex, but we are only considering these structures for the sake of simplicity. We observe here the nuclear membrane that protects DNA. DNA is coiled, giving room to chromatin threads. And also we can see centrioles. This organelle is going to play a key role during cell division, as it is going to produce microtubules that are going to move chromosomes all around. Well, back to our cell's life cycle here, we can see that after the cell is born, in this moment, it stays in a stage called G1, in which it is mainly dedicated to grow up and function, as it has to. Well, when it gets big enough, the cell has to divide. But before dividing, during this phase, into two cells, the genetic material, or the DNA that it's inside the cell, inside the nucleus, has to duplicate. In the process called replication of DNA that we have already studied, the genetic material duplicates in order to share it equally among the daughter cells. This process happens, as I'm saying, during this S phase, and is one of the most important milestones of a cell, as DNA must be kept unchanged so the descendants get an exact copy from their mother. After S phase, the cell leads to this G2 phase, in which all the structures are going to get ready for the imminent division. Cell division will happen soon after G2 finishes and includes two phases. The first one, it's mitosis, is the best known, and it consists in the division of the nucleus and the equal distribution of the genetic material. While the last one is cytokinesis, this one here, and it's mainly the division of the cytoplasm to obtain two independent cells. So let's focus then in mitosis. Mitosis is a short in time process, but however, it's complex. It assures that the DNA of the mother cell is equally shared among daughter cells, so they become clones. Mitosis has four phases: prophase, metaphase, anaphase, and telophase. These phases happen in a continuous way, so it's difficult to set the boundaries between them, but we're going to highlight the main events that take place in each one. The first phase, prophase, well, during this phase, this prophase, chromosomes appear almost fully condensed. Centrioles have been duplicated and are migrating to opposite poles of the cell, and leaving in between them a group of microtubules that will constitute what it's called mitotic spindle, which is in charge of holding, moving, and placing the chromosomes all the process through. During metaphase, chromosomes appear completely condensed, fully condensed. They are going to be aligned and placed in the equatorial plate in the center of the cell, and they are done they are located here by the spindle. Here we can see how a chromosome is held by the spindle microtubules that catch the centromere and place the chromosome in the middle of the cell. In anaphase, microtubules shorten. They become smaller, shorter, and they pull away the chromatids that build the chromosome, and they separate the chromosomes in two. Finally, during telophase, which is the last phase, the two daughter cells can be differentiated. Chromosomes decondense and turn into chromatin again. And at the end of this phase, the cytoplasm is going to divide. These two membranes are going to fuse, and the two daughter cells are going to be produced. This is it for the summary of mitosis, but before we finish, I would like you to watch the following video of a real cell's division. In here we can appreciate how chromatin is condensing, and during prophase it's becoming more clear the shape of the chromosomes. Then during metaphase they are aligning in the center of the cell, held by the microtubules of the spindle. They're moving to the center, and then chromatids are going to separate in two opposite directions, like this. While this happens, chromosomes are going to decondense and they are going to form chromatin again. The nuclear membrane reappears, and finally the two cells are going to separate. This line will divide the two cells. Okay, that's it for today. Thank you for watching, and I hope you like it. See you soon.